So there are four critical elements that we need to be familiar with in living cells. Carbon, hydrogen, oxygen, and nitrogen. They differ in their electronegativity, and they come together to form covalent bonds and other types of bonds, like the ionic bond that we discussed. It's the bonds between atoms that build molecules. And we need to be familiar with both the covalent and the non-covalent types of bonds that we see in living cells. Because again, depending on the nature of the bond, there'll be different activity that goes along with that molecule. So you can see that I've listed four different types of covalent bonds here on the slide. Remember, all a covalent bond is a pair of shared electrons between two atoms. Covalent bonds are very strong. You have to put work into breaking apart a covalent bond in a molecule. Now, I've got four names listed, and each one of these is found in a different macromolecule. You should be familiar with which macromolecule contains which type of covalent bond. The phosphodiester bond, for example, is the type of covalent bond that joins together nucleotides in DNA and RNA. Peptide bonds are the type of covalent bond that join together individual amino acids in proteins. Glycosidic linkages are the types of covalent bonds that join together monosaccharides to build larger carbohydrates, including polysaccharides. They're referred to as linkages because, unlike other types of covalent bonds, there's an oxygen atom in the center of this bond. Ester linkages are the types of covalent bonds that are found connecting fatty acids to glycerol inside of phospholipids. Four different types of covalent bonds found in the four macromolecules. Now let's take a look at the biologically relevant non-covalent bonds. Non-covalent bonds are not strong like covalent bonds are. In fact, they're often referred to as attractions. Attractions between molecules or between different parts within a molecule. The non-covalent bonds or attractions that are biologically relevant include ionic bonds, and remember those are the bonds that form between ions, hydrogen bonds, and what are called van der Waals interactions. Let's take a minute now and talk about the features of these non-covalent types of bonds. Ionic bonds form between two ions. Remember, this is the case where electrons are transferred between two atoms. One atom loses an electron, one atom gains an electron. And it solves the problem of unpaired electrons in the valence shells of those atoms. As a result of the transfer, those atoms become charged. One becomes positive, one becomes negative. So an ionic bond is essentially an electrostatic attraction between those two charged ions. Hydrogen bonds are very different. Hydrogen bonds are weak attractions between two molecules or parts of a molecule that are partially charged. Remember, partial charges form in polar molecules, and polar molecules are also hydrophilic. Hydrogen bonds got their name because one of the atoms in the bond is usually hydrogen. Van der Waals interactions are almost the opposite of hydrogen bonds. Van der Waals interactions are, again, weak attractions between two molecules or parts of a molecule that are both nonpolar and hydrophobic. So hydrogen bonds are going to form between polar molecules, and van der Waals interactions are going to form between nonpolar molecules. We can see both hydrogen bonding and van der Waals interactions in the membrane. Remember, the heads of the phospholipids will be held together through hydrogen bonding, and the tails in the phospholipids will be held together through van der Waals interactions. Water is a great example of one of those polar molecules that can form hydrogen bonds. Remember, oxygen is a highly electronegative element. Oxygen has a higher electronegativity than all of the other three critical elements that we've discussed. Oxygen has the greatest tendency, therefore, among those four elements, to attract electrons to itself. So when we see oxygen in some molecules, we know that there is unequal sharing of electrons going on. And it is the unequal sharing of a pair of electrons in a covalent bond that creates those partial charges. Polar molecules contain partial charges. And remember, we show partial charges in a drawing of a molecule using the lowercase delta symbol. Now, water is a molecule that is absolutely required in a living cell. Water has some important chemical characteristics that make it the universal solvent of living things. Water is cohesive. It has a very high surface tension, and that high surface tension comes from the hydrogen bonding that develops between water molecules. Remember, hydrogen bonds form between two polar molecules or within two polar regions within one molecule. Water is polar. Water forms hydrogen bonds with other water molecules. Water also has a couple of other critically important chemical features. Remember, the liquid phase of water is denser than the solid phase, and that means that ice floats. Remember too that water has a very high specific heat, which makes it a good insulator, and that's important in living cells. Living organisms are largely composed of water. Depending on the organism and depending on the stage of life that the organism is in, it can be made of anywhere from 50 to 75% water. All cells require water to survive. On this slide, we can see some molecular models of water and both this concept of partial charges and hydrogen bonding. On the left side here, you can see a single water molecule. Here's the oxygen in red. Here are the hydrogens in white. Here are the covalent bonds holding the molecule together. The oxygen has a very high electronegativity. The hydrogens have a very low electronegativity. That means that the pair of electrons that composes this covalent bond and this covalent bond, the pair of electrons is going to reside up closer to the oxygen and farther away from the hydrogen. The oxygen is pulling the electrons up close to it and farther away from the hydrogen. That creates partial charges. The oxygen isn't literally negatively charged, but because it has pulled the two pairs of electrons up close to it, it is relatively more negative. It is partially negative, as that symbol suggests. The hydrogens are now relatively farther away from those electrons, so they are relatively more positive. They have positive partial charges. Remember, hydrogen bonds form between polar molecules, and polar molecules contain partial charges. So when we combine several water molecules together, you start to see hydrogen bonds. The partially negative oxygens will hydrogen bond with the partially positive hydrogens. You can see a hydrogen bond drawn here as a dotted or a dashed line. That's different from that solid line that we draw when we want to depict a covalent bond. It's the hydrogen bonding between water molecules that gives it its surface tension. It's the reason that you can float a paperclip on the surface of a glass of water. So, hydrogen bonding occurs between water molecules. You can see the oxygen here, partially negative, hydrogen bonded with the hydrogen, which is partially positive. But hydrogen bonds occur in other important biological molecules as well. On the lower right hand area of the slide, you can see a depiction of some DNA. We can see adenine here, cytosine here, guanine here, and thymine here. So we have two very short strands of DNA. Remember, there is a covalent bond in this strand and a covalent bond in this strand called a phosphodiester bond that joins the thymine and the guanine together, and over here joins the adenine and the cytosine together. But there's another type of bonding that's going on here that's equally important, and that's the hydrogen bonding. Between guanine and cytosine, 3 separate hydrogen bonds can form. Between thymine and adenine, 2 hydrogen bonds can form. It is hydrogen bonding that holds the 2 strands of DNA together in the double helix. Covalent bonding is what holds the individual nucleotides together in a strand of DNA. Hydrogen bonding holds 2 strands together. Polar molecules contain partial charges. Polar molecules like to associate with other polar molecules, and they are drawn together with hydrogen bonds. Polar molecules are also hydrophilic. They are water-loving. They will associate quite readily with water because water is also polar. Nonpolar molecules do not contain partial charges. Nonpolar molecules like to associate with other nonpolar molecules, and that association comes through van der Waals interactions. Nonpolar molecules are hydrophobic or water-fearing. They will not readily associate with water because water is polar. So think about polarity as a measure of the relative asymmetry within a molecule. Polar molecules have asymmetrical sharing of electrons inside their covalent bonds. And the asymmetry comes from the differences in electronegativity. between the elements within that molecule, the oxygens, the carbons, the hydrogens, and the nitrogens. When there is a difference in electronegativity between the two atoms in a covalent bond, the sharing of electrons will be unequal or asymmetrical, and that particular molecule will be polar. That molecule will contain partial charges as a result. Nonpolar molecules are very different. In nonpolar molecules, the pair of electrons that makes up a covalent bond between two atoms is shared symmetrically or evenly because those two atoms in that bond have the same electronegativity. And that means the same low electronegativity that we find in carbon and hydrogen. So a molecule like methane, which is made up of only carbon and hydrogen, has equal sharing of electrons in all four of its covalent bonds, symmetrical sharing of electrons, no partial charges form, therefore, and we have a nonpolar molecule. The importance of polarity can be seen in the structures that are found in living cells. And a great example of this is the membrane. On the left-hand side of this slide, you can see a drawing of a plasma membrane, or a little piece of a plasma membrane. You can see it's made up of individual phospholipids that come together in a bilayer. Each phospholipid has a red head and two brown tails. You can see water depicted over here. Let's imagine that's the outside of this cell. Water depicted over here. Let's imagine that's the cytoplasm inside this cell. And we have the bilayer. The heads of a phospholipid are polar, so they will be oriented towards the water on both the outside of the cell and the inside of the cell. The tails of a phospholipid are nonpolar, so they will be tucked away in the center of the membrane. What connects the heads with the water is hydrogen bonding. Hydrogen bonding. What connects the tails with each other is van der Waals interactions. Those tails, those non-polar tails, are very similar to oil, to the kind of oil you might drop into a glass of water. And just like the oil separates itself from the water, those tails separate themselves from the water both outside and inside of a cell. That plasma membrane is what allows a cell to exist. It's what separates the contents inside the cell from the external environment. And it's made of those molecules called phospholipids. Phospholipids, because they're made of polar heads and nonpolar tails, are said to be amphipathic. That word amphipathic means containing both polar and nonpolar regions. Remember, the head of a phospholipid is composed of a glycerol and a phosphate. The tail of a phospholipid is a chain of hydrogen and carbon, along with a carboxyl group at one end. And I've forgotten to put that here on the slide, so I'll put it now. The tails of a phospholipid are called fatty acids, and they are made up of a long chain of hydrocarbon that has a carboxyl group at one end. When you attach the heads and the tails and you bring a whole bunch of phospholipid molecules together, they don't bind to each other covalently. They're simply attracted to each other. They form the bilayer spontaneously. They are drawn into this particular shape, this particular orientation, because of their chemistry. Because the heads want to be towards the water outside and the water inside, and the tails want to be away from the water. So this drawing depicts another piece of cell membrane. Only in this drawing, we're also seeing the other structures that we find among the phospholipids, the proteins, and the carbohydrate chains that create glycoproteins. Remember, the membrane is full of protein. There is just as much protein in a cell membrane as there is phospholipid. And those proteins are performing very important functions for the cell. To finish up today, I've included this table in your slide set so you can review the 4 macromolecules and their 3 levels of structure. Remember, how something is structured is going to impact the job that that thing can do in a cell. So we describe structure as having 3 levels: a very basic or primary level, a slightly more complex or secondary level, and a mature, fully formed tertiary or third order level. So in the case of protein, the primary structure of protein can be described as simply a sequence or a chain of amino acids that are in a particular order. Remember, we call it a peptide if it's a relatively short chain of amino acids. We call it a polypeptide if it's a long chain. Peptides generally have tens to hundreds of amino acids. Polypeptides have hundreds to thousands of amino acids. It's the order or the sequence of amino acids in a peptide or a polypeptide that makes each protein unique. Now, when that chain of amino acids begins to fold, it gets into its next level of structure, its secondary structure. For proteins, we're going to see both alpha helices and beta sheets forming. Twisting, turning, folding up of the chain of amino acids. Those alpha helices and beta sheets will then begin interacting with each other. Again, based on their chemistry. We're going to see hydrogen bonding. We're going to see van der Waals interactions. And we're going to get into the fully mature, fully folded, tertiary structure that we call a protein. All proteins have a 3-dimensional shape that is unique to that protein, and it is the shape of a protein that determines its function. If you change the shape, you change the function. I have similar information in this table for carbohydrates, for nucleic acids, including DNA and RNA. Notice that for all of the macromolecules listed here, the primary structure is simply a chain of something. A chain of amino acids in proteins, a chain of simple sugars or monosaccharides in carbohydrates, a chain of nucleotides in DNA, a chain of nucleotides in RNA. The only macromolecule that's not included in this table is lipid. And the reason lipid is not included is because lipids don't have these 3 levels of structure. So you should familiarize yourself with this table because this is information that you should know as we embark on our journey this semester into cell biology.